about these hydrogen bonds in terms of their bond enthalpies here. I said that they were weaker than covalent bonds, so let's look at a few comparisons here. So we can look at an HO hydrogen bond versus an HO covalent bond. And what we find is that the bond enthalpies in kilojoules per mole, it's 20 kilojoules per mole for a hydrogen bond versus 463 kilojoules per mole for a covalent bond. So we're not talking about just a little bit weaker, we're talking about much, much weaker for the hydrogen bonds here. Similarly, if we look at nitrogen, for nitrogen-hydrogen bonds, if we have an OH bound to a nitrogen, that's 29 kilojoules per mole. If it's an NH hydrogen bond to a nitrogen, that's 14 kilojoules per mole. And if we're talking about an HN covalent bond, now we're talking about 388 kilojoules per mole. All right, so again, what we see in these cases is that the hydrogen bonds are much, much weaker. They tend to be as low as 5% of what the covalent bond is. So this is actually much weaker than any kinds of bonds we have within molecules, but it's also the strongest type of bond that we can have between two different molecules. And one thing that you find, as in the case of water, is when you have lots of hydrogen bonds between molecules, it changes the property of, for example, water here. It makes the boiling point much, much higher than you might expect if you looked at the other properties, because you have to actually break apart all of these individual hydrogen bonds. And even though it's not that much energy for one individual hydrogen bond, once you get a huge number of hydrogen bonds, you're talking about huge energies here. So let's look at some examples of where we see hydrogen bonds in biological processes. First, thinking about proteins, they're absolutely hugely important in proteins. And in proteins, we're talking about a molecule that's so large that you don't just see H bonds in between two different molecules. What you actually see is what's called intramolecular hydrogen bonding, so hydrogen bonding within the protein molecule itself. And hydrogen bonding is incredibly important. It, in fact, is the shape of a protein, which I'm just showing an example here, histidine diacetylase. And the histidine diacetylase molecule actually has just countless hydrogen bonds in here, and the hydrogen bonds largely govern the shape of the molecule. So if you're thinking about any protein that has a shape, so for example, we see these ribbon structures here, they're called alpha helices. You can see some arrows which are just like a sheet structure or beta sheets. That actual shape is stabilized and formed in the first place by just many, many hydrogen bonds within the protein being in a correct orientation that allows for this particular shape to be stabilized. And we know that shape is so important when we're talking about proteins, and that's because the actual shape of the protein governs the interaction with other proteins or other small molecules. The shape of the protein is very important in terms of positioning the molecule or the specific atoms that are involved in the chemistry that the enzyme carries out being in the correct position. So hydrogen bonds are very important in terms of proteins. We can also think of them in terms of sugars or polysaccharides. This is a nice dramatic example. Thinking about the structure of a protein is a very microscopic way of thinking about hydrogen bonding. If we want to take it to the macroscopic level, we can talk about H bonding in trees. So if we're talking about trees are made up, a major part of a tree is the polysaccharide cellulose. So that's shown right here. Cellulose is a chain of glucose molecules linked together. It can be as many as 1,000 or more glucose molecules linked together covalently.